Welcome everybody to Supplementary Video 27.1. This is the first video that we have covering the reproductive system. You'll notice that this video is going to cover a lot of male anatomy and physiology that gets left out in the high school portion of the side. We don't have a lot of extra over the course of this unit that I'm going to add to on the female reproductive side just because I'm already adding in so much there. But we are going to get really in depth into pregnancy and some things like that associated with development of the fetus. So this video is on, in addition to the videos on male anatomy here, I leave out the muscles associated with the scrotum. So first you have the dartos muscle. It's going to be the most superficial muscle here. You can see how it is going to wrap around the superficial side there. It's also going to invaginate and create the scrotal septum to separate each testis into the left and right. You also have two cremaster muscles that are going to be descending from the internal oblique that are going to be responsible for holding each. You can kind of see how they're sort of net shape. And from the descent of the testes, they originate near the kidneys, but they're actually going to descend down into the scrotum. They're going to push through that internal oblique. They'll be caught by the cremaster muscles, but that's a huge reason why males are much more likely to suffer from inguinal hernias or any sort of hernia, is because that internal oblique has been really weakened over the course of that male development. The testes have two layers of tissue that I don't cover super in depth. I just mentioned one of them. First, you have the tunica vaginalis. It's going to be a serous membrane, so think back to our epithelial unit. And it's going to have a parietal and a visceral layer. And the tunica albuginea is going to be a dense connective layer. It's going to divide each of the testes here. So you can see this tunica albuginea and how it's going to be dividing each into the lobules that we see. Now, each testis has about 300 to 400 lobules that are inside each of those lobules are the seminiferous tubules. And within those seminiferous tubules are going to be the Sertoli cells and the germ cells, which we'll talk about on the next slide. But you can see the tunica vaginalis, how it's going to be wrapped around. It does have that parietal and the visceral side, and then the tunica albuginea responsible for separating each of the lobules. So the seminiferous tubules have those two types of cells, those Sertoli cells and the germ cells. The Sertoli cells are going to be support cells. They're going to be secreting different molecules, different chemicals to support sperm production. They'll wrap around the germ cells to create a blood-testis barrier, sort of like you have a blood-brain barrier. The Sertoli cells are going to be responsible for keeping the germ cells separate so that you don't have any sort of autoimmune response because they aren't getting into the rest of your normal circulatory system, and so you aren't having any sort of foreign antibodies or foreign antigens being recognized that are going to cause a response there. So those Sertoli cells are really important for keeping those separate. The germ cells, it actually has 5 different types of cells, but they each represent a different stage of spermatogenesis. So that's why I just leave them as germ cells here. You're going to start with spermatogonia, which are the stem cells. So everything is going to originate from a spermatogonium. This IUM, that is the singular version, IA is the plural version. From there, it'll go through mitosis. You get this primary spermatocyte. One of them will revert back to being a spermatogonium. The other will be destined to become the sperm at the end here. But you go through meiosis 1, you get the secondary spermatocyte. You go through meiosis 2, you get the spermatid. And then you go through spermiogenesis to basically get the fully formed sperm that you have here. Originally, you're just dealing kind of with this 2N is your normal amount of DNA. Now we have the DNA split, so it has half the number of chromosomes that a normal body cell would go through meiosis 1, meiosis 2, but it's just dealing with a cell here with the correct amount of DNA that you need in those spermatozoa, but it still needs to go through spermiogenesis in order to get the tail and get the acrosome and the cap and things like that. So here's a cross-section of a seminiferous tubule. You can see the lumen, the gap in the middle here. Everything is going to be originating from the basement membrane here and working its way towards the lumen. Okay, one last thing I want to add is the testosterone feedback mechanism. Obviously, that's going to be very important in males as they develop not only in utero, but through puberty. So testosterone is going to originate or the control for it, I should say, is going to originate in the hypothalamus when you have this gonadotropin release hormone. It'll stimulate the anterior pituitary to release follicle stimulating hormone and luteinizing hormone. Hopefully, you remember these abbreviations from the nervous system unit. So the anterior pituitary is going to release FSH and it's going to release LH. This FSH is going to go to the Sertoli cells, going to go to the to create ABP, this androgen binding protein, which is going to release testosterone. The LH is going to go to the Leydig cells, and that's going to stimulate testosterone release. So this here, you have this mechanism here. You do have inhibin present, as well as just the level of testosterone is going to be really important. So as you get increasing levels of testosterone, that is going to inhibit your release of GnRH. And so if you don't have any GnRH being released, you don't have any FSH or LH being released, meaning you don't have testosterone being released. So when testosterone is at high enough levels, it's actually causing less of it to be formed, saying, hey, we got enough, we don't need to make any more. Over here on this side of the feedback mechanism, you have inhibin here, which is going to negatively feedback to the anterior pituitary to inhibit FSH. And so if you're inhibiting FSH, you don't have any of this ABP, you're going to be inhibiting the testosterone release here. So overall, this is a negative feedback system. You produce more, and that's going to cause the system to shut off. But if you have less, that's going to cause this system to create more to get it back to its normal levels. So remember, negative feedback systems, they want to return you to normal levels. So if it's low, it's going to stimulate the production to increase it. And when that level is high, that's going to inhibit the release of testosterone here. So this is a very classic negative feedback system. So that is the end of our first video, supplementary video 27.1. I think this is the only one that we have for males, and so we have a little bit of a break before we get into the next one, talking a little bit on female anatomy, but then really getting into pregnancy and development.